All right, here we go. Yep. Okay.
No, yep.
Yep, I recognize that name. There you go. It's all good. Okay. There we go. There we go. Okay. So thank you for joining us, everyone. We'll give people a few more minutes to log in, make sure we're not missing anybody. Right? Right. Okay. Yeah. I know. If you go to sleep, I promise you'll feel so much better. I promise. Yeah. Okay. Well, that's why I said sleeping was a good idea. Okay. So for those of you who are joining us, we won't start quite yet, but I just want to introduce myself. My name is Mirka Zaplatow, and this is my noisier companion here, who is absolutely resisting bedtime. I'm the Director of Education at the McAuliffe-Shepard Discovery Center. Thank you so much for joining. It's you, kiddo. You're yelling at you. For joining us tonight for our Superstellar Friday event. I feel like we're getting to the point where we're kind of knowing what we're doing with this, which is fantastic. And we're trying to bring in a wide variety of STEM topics so that we all learn a little bit. The way this is going to work, if you haven't joined us before, and I apologize for the noise, this isn't normally the way things go, is that Wallis will be giving her presentation and I will be monitoring the chat and the question and answer, and also if anybody's raising their hand. So at regular intervals, she'll stop and ask me if there are questions, and I'll feed any of your questions to her. So if you have a question, you can write it in the chat box. You can put it in the, I know you're tired. In the Q&A, and the Q&A option, if you see down at the bottom of your screen, there's a place to get into the question and answer. That does give you the option to see other people's questions and vote for them and say, yes, this is an important question. But you can also raise your hand. Right now, you're all muted. So if you raise your hand, what will happen is I'll say so-and-so has a question, and then I'll unmute you and say, you need to also unmute yourself to ask your question. So you will be able to talk if that's the way you'd prefer to do it. And we'll just kind of go through it, stopping periodically. So let's see where we're at, 7:02. So Wallis Boram is one of our newest employees, originally from the West Coast. She's now hanging out in the Northeast, just in time for winter. So well planned that way. A recent graduate of Bard College in New York. She's joining us as the education specialist to work on a specific grant project where she'll be working with high school students across the state to help them create a traveling exhibit, which is pretty exciting. But what I find really exciting is that tonight she's going to be talking about her thesis work with a species of worm that I didn't even know existed until she told me she had done research on it. So I'm going to mute myself. I'll be here to check on your questions and pass everything along, but I'm going to turn it over to Wallis.
Yep. All right. Hi, everyone. I'm Wallis, and I'm going to start screen sharing so that you can see the presentation. All right. Okay, let me get this. All right. So, this is the title of my presentation, Invasion of the Jumping Worms, because what I'm going to be focusing on this evening is Asian jumping worms, which are a fairly new invasive species to the Northeast. But first, there we go. A little bit about me. So like Mirka said, I just graduated from Bard College with a bachelor's degree in biology in May. And this was a pretty crazy time to graduate, but I mean, it's a pretty crazy time to do anything. So when I first came to Bard 4 years ago in August of 2016, I thought that I was going to be a chemistry and written arts double major. That obviously changed, but I decided to be a biology major because I found that as I was taking chemistry and written arts classes, I was kind of just skating by and realizing that I wasn't putting a whole lot of effort into the classes because I felt like I just knew how to get by with them. And so because I was on the pre-med track sort of, and that obviously didn't happen, I took a biology class in my freshman spring, and it was a genetics course. And for the first time in my first year of college, I felt really challenged. And so despite genetics not being my thing at all, in fact, we had a very strenuous relationship, I still decided to change my major to biology. And over the course of my courses at Bard in the past 4 years, I developed a love for ecology and conservation. So that was my specialty when I was working towards my biology degree. So here I have a few photos. On the right is me in my field ornithology class, doing field work, going out and doing bird counts. And I'm actually standing on the frozen Hudson River, which was pretty cool. Also a little nerve-wracking. Had a friend right there to pull me out in case I fell, but you know. And then to the very far left is me wearing waders, getting ready to wade into the bit of water you see just to the right of it. And that is Kruger Island Road, which is a walkway that goes to Kruger Island. But obviously it's not much of a walkway right now because since the Hudson is tidal, the area goes through high tides and low tides. So I was doing bird counts there for my field ornithology class at 6 in the morning, and some mornings I just had to put on the waders and go trek out there. But it was a great way to start my morning, and this class was what made me realize that for the rest of my time at Bard, I did really want to do field work-based ecology. And then the two photos in the middle are just showcasing how beautiful I think Bard is and why I wanted to come back to the Northeast after I graduated and then returned home to Seattle for about 7 months. So my time at Bard, though, wasn't just about being a biology major. One of the most influential experiences that I had there was for 4 years, I was a member of Bard Emergency Medical Services. So I was a New York State registered EMT. So in the top left, that's just a picture of me and the Friday night squad on Spring Fling, which is Bard's big, essentially party weekend. So we were pretty busy that night. But what Bard EMS serves to do is that in the relatively rural setting of, well, where Bard is in the Hudson Valley, sometimes it can take about 20 to 40 minutes for an ambulance to arrive. So Bard EMS had a response time of about 3 minutes. So if someone needs an ambulance, we were there to keep them stable until the ambulance showed up. But we ourselves were not a transport agency. I also had the opportunity to go study in Oaxaca, Mexico for a month in my junior year. And that was a totally spontaneous decision to take an intensive Spanish course after never taking a Spanish class before. I actually took Chinese in high school. So this was one of the places we visited. It's Monte Alban, which is a ruin where the Mixtec and Zapotec people lived a long time ago. Around 10 BCE. And then, so that was an incredible trip. And that's actually us standing in front of the widest tree in the world. And then I also had the opportunity to take an air, earth, water class, which looked, sculpture class, which looked at waste in the air, in the water, and on land. So it was a nice meshing of both art and science. And so that's a sculpture that I made that looked at climate change specifically, and what would happen when sea levels rose, and so we put our sculptures in ice. So all of these experiences kind of, led towards a culmination of my senior thesis, which I'm obviously going to get into, and just my love for nature and ecology. And what made me realize too, especially through my ecology classes, was that even though it was my field, science can be really inaccessible. So I knew that when I graduated, rather than becoming a career scientist, I wanted to go into science communication and informal education and make science as accessible to all types of people in all types of communities. And so here I am. So yeah, does anyone have any questions?
Nothing at the moment,
okay.
But if anything comes through.
Okay, cool. Sounds good. Then I will keep going. All right, so time to jump into the jumping worms. So on the left are jumping worms, and jumping worms are a type of earthworm. Typically, what you think of when you think of an earthworm is a European earthworm, which is also an invasive species. So every single earthworm in the Northeast is invasive. And this is because after the last glaciation, all of the native earthworms were wiped out, and European earthworms came over in the 1700s with European settlers. But much more recently, Asian jumping worms coming from East Asia arrived in the mid-1900s, actually in New York, in both New York City and the Hudson Valley. And so for several reasons, jumping worms are much more aggressively invasive than those hailing from Europe. And so there are a couple of key differences for how you can identify a jumping worm versus a European earthworm. So the most distinguishing one is the clitellum, which is the that little band around the worm that we typically think of as kind of looking like a Band-Aid. So European earthworms have a raised red, brownish, orangish one, versus jumping worms have a flat, sort of milky colored one that lies flat against the rest of the worm. Another difference is where they live in the soil. So jumping worms live in the very top layer of the soil, in the leaves that have fallen, as well as in the top 10 centimeters of soil. So that makes them something called epiendogic. Versus European earthworms can be epigeic, endogic, or anesic, which means that they either live in those leaves that have fallen, which is epigeic, and that's where the epi of epiendogic comes from. Endogic, which is again the top 10 to 20 centimeters, or anesic, which is burrowing straight down into the earth. And because of where jumping worms live and the way that they move, they create these this soil that looks like coffee grounds. So you can see the difference between the European species on the right versus the Asian jumping worms on the left, and how with the European species, the soil is kind of clumpy and sort of wet looking still, versus with the jumping worms, it looks very dry and it's really clumpy, and that's due to the way they move as well as their castings. And they also have an annual life cycle, which is pretty unique for worms. And so by annual life cycle, think of a weed like a dandelion, where it grows throughout the summer, reproduces, and dies in the fall. So the jumping worms lay their cocoons, and then the cocoons hatch in the spring. The adults come to maturity at the end of the summer, die in the fall when it gets to be too cold, and then as soon as temperatures rise again, the cocoons hatch and it starts all over again. So as I mentioned, they have a pretty unique style of moving. So I'm going to show you a video of what that looks like.
Wow, look at that guy go.
Okay, so that was, that was one day that I went out into the field with my advisor to collect and look for worms, and is a great example of how those move. They're called jumping worms, crazy worms, snake worms for a reason. So so invasive species. These worms are an invasive species, but why do we care what an invasive species is? So first, invasive species are harmful invaders of ecosystems they are not native to. However, that doesn't necessarily mean that if a species is non-native, it's invasive. There are non-native species that are not necessarily invasive. And invasive species are often spread through human activity through a variety of reasons. So for plants, a lot of the time, plants are introduced for decoration purposes, people just thinking that, oh, this plant is beautiful, I want it in my garden. And then it becomes an invasive species and takes over native ecosystems and chokes out the other plants. And then a wide range of other species are actually hitchhikers from global trade and transport. So think of rats sneaking onto ships and coming onto islands where they've never been introduced before, and then decimating the local species there. And so invasive species that are introduced have a lot of impacts on the ecosystems that they find themselves in. So they can alter the structure of the species that are in the ecosystem, and so they change the way the ecosystem functions. They also alter how many types of species of plants and mammals and birds and insects and etc. Are in that ecosystem. And they also change the way that, so that they can change the diversity of an ecosystem, meaning that, so say you have 10 species and each species has a certain number of individuals in it. When an invasive species is introduced, you could still have those same 10 species, but instead of there being multiple in each, it could drop down to one, and then there could be a lot of individuals in the invasive species. So that's also referred to as richness, which is how many are in each species, how many individuals. So although it looks as though there's a fine amount of species and that nothing has changed, there's actually quite a big imbalance. So the title of my thesis, and this is a lot to unpack right here and quite a mouthful, was Effects of Amynthus and Metafire Jumping Worm Invasion on a Northeastern Temperate Deciduous Hardwood Forest Ecosystem. So essentially what that means is that I wanted to look at how jumping worms affected forests around Bard and in the Hudson Valley through both their effect on plants as well as their effect on the soil. So jumping worms have a few effects, some on plants, some direct, one direct, and a couple indirect. So directly, they can affect seedlings by consumption of the seedling. They are known to actually munch on the plant. And also the way that they move, that wriggling motion in the soil, can disrupt the roots of the seedling. They also can change the nutrients in the soil. So OM stands for organic matter, which is the leaf and organic matter debris that you see on the floor of a forest. And that organic matter, those leaves and plant debris, when they decay, they release nutrients into the soil, and those can be plants can reuptake them and use them to grow. So common ones that plants need are nitrogen, potassium, and carbon. So when jumping worms completely deplete or nearly completely deplete the organic matter, those nutrients can be lacking in the soil. They also potentially affect bacterial to fungal ratios in the soil. So in a healthy forest, what the understory plants want is a higher ratio of fungi because of mycorrhizal fungi, which creates sort of these tubes between the fungi and the plant in order to help the plant get more nutrients. But when there is more bacteria, that connection can't really be made, and so the plants lack an ability to get nutrients in that capacity as well. So if there was a higher bacterial ratio, if there was more bacteria, say it's the orange triangles, you would see maybe 5 orange triangles and 3 blue circles, versus if there was more fungi, there would be, say, 5 blue circles if the fungi were the circles and 3 orange triangles. Now these all have sort of a flowchart effect that then affect plants as well. So again, there's the direct relationship between the worms damaging the seedlings, as well as changing the bacterial to fungal ratio, which affects the soil nutrients that the plants can uptake, as well as the jumping worms directly affecting the soil nutrients and also then the nutrients that the plant is able to reuptake. So again, I wanted to see how jumping worms affected plant productivity and forest soil. And by plant productivity, that means how much a plant has grown in a certain amount of time. And so how I measured that was after 2.5 months, I harvested the plants, dried them, and weighed them. So my prediction was that after a short growing season, which again was 2.5 months, there will be an effect of jumping worms on plant biomass and soil nutrient composition and bacterial to fungal ratios. So I did this in, as I mentioned, in Northeastern deciduous hardwood forest, one at Bard. So this is an example of what that looks like. It's made up of a lot of maples and oaks. And this is where you can find jumping worms. So as I mentioned, they were first introduced in New York in the Hudson Valley and close to New York City, which is right there in the center of the map. And the yellow is Metaphyre hilgendorfi, which I think is maybe the coolest scientific name for a species that I've ever heard. My second favorite is for the American bison, and that's bison. But then the orange is Amynthus agrestis, and the green is Amynthus tokyoensis. And so these are the 3 species of jumping worms that were all introduced at the same time and also seem to co-occur. So from first being introduced in New York, in the past 10 to 20 years, they have expanded both northward, westward, and southward a bit. So there also is they have a big presence in the Midwest as well. That was from transportation from New York to the Midwest that they spread there. And there's been a lot of research done primarily in Wisconsin on the effects of jumping worms. So yeah, this just shows where they were introduced and now how far they've spread. And this also indicates that they can spread very far in a short amount of time, which is one of the reasons why they're a big threat to the forests up here. So now getting into what I did, this on the right, or sorry, on the left, is a map of where I did my soil collection as well as jumping worm collection. And so I'll explain what that means. So for my experiment, I wanted to collect soil that had never been exposed to jumping worms before. So I found a plot of forest that didn't have jumping worms, collected it, put it into those tubs down there that you can see on the bottom right. And that was the basis of my mesocosm, which mesocosms are essentially mini ecosystems, and I'll talk about those more in a second. And then I also had a jumping worm collection site, which is where I collected all the worms. And so the top right was a plot I created in order to sample the worms. So by creating this plot, you can get an estimate of density. So how you typically sample for worms is you take a jug of water and mix in 40 grams of mustard powder to make mustard water. Then you create the plot. You pour the mustard water over the plot, and as the mustard water sinks into the soil, it irritates the skin of the earthworms, and so they all rise to the surface, and it's really one of the craziest things ever. But I found that actually a better way to find and sample for jumping worms, because they live just in that top layer of leaves, dead fallen leaves, you can just sweep away those leaves, the organic matter, and find worms just there wriggling on top of the soil. The most I ever found in a plot about that big by just sweeping the leaves away was probably about 25. And those things can get massive. And I also forgot to mention that another way that you can identify them is that they have a slight iridescent sheen. So there's nothing like picking up this shiny, rainbowy, huge, wriggly worm. And they also, as a defense mechanism, will drop their tail if they feel threatened. And so when I picked up, it was so huge that when it dropped its tail, I actually felt the like snap of it coming off. So yeah, they are huge and can exist in really dense populations. And so that's another reason why they're so invasive, is because they don't need a whole lot of like space to have a really high population. But okay, anyway. So here is my experiment all set up. These are the mesocosms I was referring to. So they're made out of bus tubs, as well as wooden frames that I created and mesh encircling the whole thing, because, you know, jumping worms, they do jump. And so I learned that you actually do need to enclose them in mesh, otherwise they will escape. And of course, Bard is just in the middle of the forest. I did have issues with herbivory, and multiple times had to chase away groundhogs and deer who were coming to snack on my mesocosms and the plants within them. I also then had a tent over everything that served to not only protect from the insane August Hudson Valley storms, but also to mimic forest canopy. So I actually took a measuring device, and, you know, although it wasn't a perfect comparison to the forest canopy shading, it got close enough that it was a nice comparison. And then 8 mesocosms had jumping worms, and 8 didn't. And so the ones with jumping worms are marked with the orange duct tape, and the ones without have no duct tape. So within each mesocosm are native or naturalized understory plants. And what I mean by naturalized is that although they are not native, they're also not invasive, and they've been here for a long time. Then I also had the field soil I showed you a picture of the leaf litter that was on top of the field soil that I also collected. And then, like I mentioned, the mesocosms with worms or without worms. So this is what the mesocosms look like when I first planted the plants. There's a nice little community in there of plants. There is a fern that you can see right there, 2 types of naturalized vines, bunchberry, phlox, and foam flower, which that's the big sort of like large leaf one that you see right there. So I planted these, waited 2.5 months, or put in the worms, waited 2.5 months, and kind of let them take the reins and do the science. Watered them about once or twice a week, moved them around, you know, kind of let them do their own thing. And then after all that, this is what the plants looked like after, what the mesocosms look like after. So they really took off, and I was excited to start harvesting them, to weigh them and get my first results. So things in science always go wrong. It's just inevitable. And so something went wrong pretty much immediately when I started collecting my data. I started harvesting the plants, and like I mentioned, you have to put them in a dryer, not like a laundry dryer. It's specific for drying plants, because you have to dry plants before taking their weight to measure their growth, because different plants hold different amounts of water. So I put them each species individually in their own bags, stuck them in the dryer, went home for the weekend because you can leave them in there for 24 to 48 hours. And then on Sunday night of that weekend, the science building, the laboratory coordinator got a call saying, "Craig, you need to come to the RKC, the science building, right away. The biology lab is filled with black smoke. The fire department is on its way." So I set the science building on fire, which I think is really just, not many people can say that, I think. But I lost 2 samples. Fortunately, it was one of the plants that were in a mesocosm with worms and one in a mesocosm without worms. So it balanced out, but it meant that I only had 7 samples on each side of my plants. And then this is one of my favorite track changes on Word that I've ever received from my advisor, Kathy, when I tried to explain in my thesis why I all of a sudden only had 7 samples in my worm treatment and 7 samples in my non-worm treatment. So, but I still had results. So let's talk about those. What I first wanted to know was how jumping worms affected the overall biomass of plant communities. So the overall growth of the plant communities, how much they grew during these 2.5 months. So I saw that there really wasn't a significant relationship. And when I say significant, I mean that there wasn't indication that this happened in relation to each other, and that it was so significance is 0.05, which means that there is only a 5% chance that this happened randomly. Anything above that means that this might not have happened randomly. We don't know, so we can't say that there is a direct relationship between these 2 things. So based on my results, I couldn't say for sure if there was a direct relationship between overall plant growth and whether there were worms or not in the mesocosms. So after this, I wanted to see how the worms affected the individual plant species between the treatments. So again, I have the 7 species down there below, and most of the species weren't affected except for one, Tiarella cordifolia, which is the foam flower. So I saw that there was actually an apparent relationship between mesocosms with worms and mesocosms without worms, and the growth, the biomass of the foam flower. So the p-value was 0.04, which just means that there was a low likelihood that it happened by chance, and probably there was actually a relationship. So now the next thing that I wanted to look at is diving into the soil aspect of the experiment. So first, I wanted to look at the organic matter, how they affected the leaf litter. And because that's a big part of what they do to ecosystems to change them, is that they completely just decimate the leaf litter, as you can see. So the one on the left is a mesocosm without worms, pretty much identical to when I put it in there, versus on the right is a treatment with worms. And I did not clear any of the leaf litter out. That's exactly what it looked like when I harvested the plants, and then I took a picture of it. And you can also see this is a great example of the sort of coffee ground-like texture that the jumping worms create in the soil. So I found that there was a relationship between the worms, no worm versus worm mesocosms, and the percentage of organic matter. So, I mean, even visually, you can tell that they just really chomped all the way through that, all those leaves. So next, like I mentioned, I wanted to see how the worms affected soil nutrients. So originally I had thought that just like this figure, I could graph soil nutrient and then compare them in non-worm versus worm treatments. However, there just was, it was just embarrassing how much that just didn't happen. There was no relationship between that. So I decided to take a different approach and try a principal component analysis. And essentially what that does is it takes a few variables and squishes them down into just like one point where you can visualize all of it happening at the same time. So with a principal component analysis, what you're looking at right now is a 2D image, but imagine that you are standing in a corner with a wall on either side of you, and you are looking out into the rest of the room. You would almost see those dots floating in the center of the room in a three-dimensional space. So each dot represents one mesocosm, and the darker dots are mesocosms without worms, and the gray dots are mesocosms with worms. So right off the bat, you can tell that all the dark, no worm mesocosms are pretty clumped together. And that's exactly what I wanted to see, because it means that all of the soil nutrients, their composition and the amount of each nutrient in the soil was very similar. So that means that the soil that I collected, the soil that had not been invaded by jumping worms, was actually never invaded and was an accurate control. Versus the light gray dots are mesocosms that had worms introduced. So although I didn't see that there was a relationship between the jumping worms affecting individual values of nutrients, they did really affect the composition of them. So when combining all the nutrients into one sort of variable, they varied greatly, because some mesocosms could have had higher carbon than others, or higher nitrogen, or higher potassium, and then some could have lower nitrogen. And so that's why that variability looks like that. It's not that each nutrient is affected the same, it's that they're affected in different ways. So there are a couple reasons why the jumping worm mesocosms, these values could be all over the place. It could be that based on the way that the worms moved and their activity, they could have affected the soil in different ways. And also because I wasn't able to go in and every single day count how many worms were still alive. I put 25 worms into each worm mesocosm, but there's a big possibility that some of them just died and then weren't contributing as much to the effect of on the soil nutrients and their composition. So here, this is a big old table, pretty much all that I wanted to do here. Don't even need to really think about the tests or the statistical value, but I just wanted to show that because I mentioned that I was going to look at bacteria and fungi. There obviously isn't a graph of that, and it's because I just found nothing. There was no significance in any capacity, no results even worth sharing. So it could be that they just don't affect bacteria and fungi in that capacity, or it could have been that my experiment just didn't run long enough. But I mean, you know, this went against my prediction, but the point of science isn't to go into an experiment knowing exactly what you're going to find out. So, you know, it didn't meet my prediction, but that's okay. I just move on and try something else. So in sort of conclusion, jumping worms didn't affect total plant community biomass, so there wasn't a significant relationship between worms versus no worms and the overall growth of the plants in the mesocosm. But they did negatively affect the foam flower and caused decreased growth in the mesocosms with worms. As well, they negatively affected organic matter. So as you saw, they just ate all of it and it was completely gone. And they caused variability of soil nutrient composition. So with all of this in mind, some explanations and some implications. The loss of organic matter that these worms create means that the decaying leaf litter doesn't redistribute nutrients in soil. So as I mentioned, that can be pretty consequential for plants because they need those nutrients in order to be productive and grow. And so this creates both short-term and long-term consequences. In the short term, the plants currently in the understory lacking these nutrients are going to suffer and they're going to grow less and they're going to reproduce less. So this then creates the long-term consequences of potentially a species just disappearing from that ecosystem. And in its place, actually jumping worms, I observed, but this wasn't part of my thesis. In places where there were jumping worms, there was also more incidence of another invasive species, a plant invasive species called Japanese stillgrass. So when one invasive species comes in and changes the ecosystem, it also opens up the opportunity for another invasive species to come in. And then I also saw that changes occurred rapidly following this simulated invasion of jumping worms. So again, my experiment only lasted 2.5 months, which on the grand scale of everything is a pretty short amount of time, and yet I was able to see some preliminary results of what these worms can do to ecosystems. And so finally, some takeaways from all of this. My thesis didn't serve to propose any sort of solution to what to do about jumping worms. It just served to give more information because since they're so new, this was a pretty novel new study, and there hasn't been a lot of studies or research done on them, especially on their effect on plant growth itself. A lot of more research is on the soil, which is obviously very important for the plants, but not many people have looked at the actual growth and productivity of plants. So I'm adding more research to this, but again, no really solution. And right now there unfortunately isn't a solution. One proposed way to manage the worms is that cocoons can be destroyed under extreme heat. So prescribed fire is an option, however, that also still destroys the forest. So that's not a great solution. Another issue is the relationship between jumping worms and climate change. So cocoons hatch when there is a sustained temperature of 10 degrees Celsius as it's transitioning into spring. But as spring temperatures get warmer and warmer earlier and earlier, this means that the cocoons will hatch earlier. And same with in the fall with warmer temperatures sustaining. I mean, it was like what? It was like 68 degrees here today. That's crazy. So with warmer temperatures going later into the year, the adults can survive longer. So that gives a longer window of maturity, which means more time to reproduce, as well as more time to just mess up the forest even more and cause more havoc. So yeah, oh, and another really interesting thing, especially because I was doing this research in the Hudson Valley, is that there's an interesting relationship between jumping worms and ticks, or potentially an interesting relationship. And that is because jumping worms consume the leaf litter, very much so deplete it, but ticks live in leaf litter. So, you know, it's kind of a hard thing to think about because yes, the jumping worms are consuming this leaf litter and having a bad impact on the forest, but if the leaf litter is gone, and that could potentially mean a lower tick population, then that could lower incidences of Lyme disease, which is one of the, you know, like leading diseases and causes of death in the Hudson Valley. So yeah, it's a complicated topic, not really any answer about what to do on any front, unfortunately, again, but yeah, just this is my little take on these worms. So thank you.
So if anyone has questions, you are welcome to put them into the chat box, raise your hand, or put it into the Q&A. I see that somebody's got their hand up, so let me see who that is. Okay, George, I see that that's you. So hold on just a second. I'm going to. Sorry, I just totally muted myself while I was doing that. I apologize. So George, I think I just unmuted you now. So you should be able to unmute yourself possibly and say something.
Okay, thank you very much.
Yeah.
Thank you for the presentation. It was very interesting.
Good.
I guess my question is, throughout our life, we're told that worms are beneficial to us. Mm-hmm. Now we're hearing something different about these types of the Asian jumping worm. Mm-hmm. Are they taking over our normal worm population?
Yes, so there is some research saying that they are dominant over some species of European earthworm. Um, so yes, however, an interesting thing is that um, earthworms that we know of that were here before Asian jumping worms and that we're all really familiar with are great in cases of agriculture and compost and fishing. However, they're still pretty detrimental in some ways for native ecosystems. So yes, you're right that in a lot of ways, typical earthworms, or what we know of as typical earthworms, are great, but they still pose some issues. However, these jumping worms aren't helpful at all for composition, or sorry, composting, aren't helpful at all for agriculture. In fact, not helpful at all based on what's proposed about them. And you actually can't even fish with them because of how much they wriggle away. And that's one of the theories for how they became widespread invasive in the first part, or in the first way, was because people tried to use them to fish, but then they wouldn't stay on the hook. And so fishermen just dumped out the cans of them. So.
And can I have a follow-up question?
Yeah, yeah.
What is meant by that they reproduce in large numbers?
Yeah. Oh, yeah.
There's a life cycle to them, correct?
Mm-hmm.
But once that life cycle is over, it's over. But do they reproduce, keep reproducing more after that?
So they reach maturity, and then in the fall they lay cocoons, and so those cocoons, even though the adult worms die, the cocoons then can survive the winter and then rehatch in the spring. So as that population grows over time, and especially with climate change, as the worms have more time to reach maturity and reproduce, then those population numbers are going to skyrocket. And for some reason, jumping worms are able to live in a much more dense population than other worms. So again, like I said, in that small little plot, I found maybe about 25 of them, versus with a typical European earthworm doing that, I may have found like maybe 10 or something.
Wow.
Like.
Wow. So it is a ratio of like 2.5 to 1 then.
Yeah, something like that. That's not a direct number, just observational, because I wasn't counting and sampling European earthworms, but there was a noticeable difference between the number of each.
And I promise this will be my last question.
No worries.
Are they found only in the forest, or are they in our backyards?
They're in backyards. A big issue is that the cocoons can get into mulch and can get stuck in boot treads, so people can walk around and take them with them, or buying mulch, trying to mulch their garden, inadvertently put jumping worms into the garden. And actually, one issue that I had when creating this experiment was going to buy native plants, going to plant them, and then realizing, oh, there are already jumping worms in this like little container that I bought the plant in. That's not great. So they're everywhere. And I mean, the Cornell Cooperative Extension in Dutchess County, which is, I worked with them a little bit to when I was first getting started, to help me identify the worms and where to find them. They told me that they had to actually cancel their annual like little plant sale thing that they had, horticulture event, because they just found jumping worms everywhere.
Wow. What does a cocoon look like?
It's just a little tiny ball. It's so hard to see. It doesn't look like anything really, but it's round and it's small. I can maybe pull up a picture really quickly. So they look like that. Yeah, so they're very
Oh
small.
my gosh. Wow.
Yeah.
Yeah, look at the size. Mirka, thank you very much, and thank you for the presentation.
Sorry, I apologize for the noise in the background. He's pretty excited about something right now. We had another question, which I echo as well. How do we know how they first arrived here?
So a little bit unclear. Again, like I said, well, probably because it was in the 50s and they were introduced in New York, I would assume that it was through global trade. So I probably, there's not a whole lot of evidence for what exactly happened, just as what we assume with lots of invasive species. Obviously, like plants, they weren't brought here for an ornamental purpose, but I'm guessing it was more like, although they could have stowed away in plants, which is a big possibility, or bringing over agricultural supplies, or just even, again, getting stuck in people's boot treads. So there's really no correct answer, at least that anybody knows of, but it could be from a variety of reasons.
Okay, because I had always heard that European earthworms, for example, made it here because they were in the dirt that was in ships in the ballast. And so, they weren't intentional, they were stowaways. And I same thing.
Yeah, no, they were stowaways as well. Yeah, they weren't introduced on purpose, because, sometimes species can be introduced on purpose. Sometimes, interestingly too, like this is one of the most interesting reasons invasive species are introduced, at least for me personally, is to control another species. So what was it? I think it was the brown tree snake that was introduced to manage the rat population on, I think, Guam. Is that what it was? I don't remember.
And went on to eat all the...
Yes. Yeah. Which I actually interviewed. This is totally off topic, but I actually interviewed an ecologist who did research on seed dispersal in Guam, and how crazy the islands Guam looks compared to another island based on that. But anyway, yeah. So I just find invasive species really interesting. So it was great that I got to do this project.
Okay, and we had another question as well, follow up from George, about if they had any positive purpose. So I know you mentioned, is it good or is it bad, the ticks thing? Yeah. I didn't know maybe where they're originally from, if they're, a bit more positive in their roles.
Mm-hmm. Yeah, that is actually something I don't know about because they are originally from East Asia. I'm not sure what those positives they could potentially have are there. That's not something I looked into, but that is interesting, and I should look into that. But here, yeah, I mean, as far as we know, it seems like they can't help in composting and agriculture like European earthworms can, and that right now it's only possible that potentially they beat out in some capacity other harmful like things that they interact with, like ticks. And they don't not to keep adding to the list of how bad they are, but they also don't just affect plants. Obviously they, like I mentioned, affect ticks, but that's more indirectly. But they do outcompete native millipedes. So they are directly affecting other invertebrates as well.
And maybe this is beyond the scope of your talk too, but where they're from in East Asia, sorry, he's so loud. Is that a similar habit? Like, is it a forested space like us, or are they more rainforest, or do you know?
Oh yeah, I unfortunately don't know that as well.
Okay, I was just curious if
yeah.
maybe breaking down the leaf litter more quickly would be helpful if they were in a certain spot or something like that.
No, that is a really interesting. Yeah, All right, I have more to look into this. I thought I finished my senior thesis in December, but I'm just carrying it with me.
It never is. There are always questions.
Yeah,
If anyone else has any questions, please either raise your hand or type it into the chat or the Q&A. I have to say that there are a number of moments in your talk where I was like, oh, I had no idea, or well, that's an interesting thing right there, because I mean I saw on the map that there's a spot in New Hampshire at least, or right on the New Hampshire-Vermont border, and I'd never heard of these until you mentioned them, so I was totally surprised to see where they were.
Yeah, no, and it's really interesting how those dots that they show are not just like, oh, they're found in this sort of general widespread area. It's like the way that they spread, they're very localized. So the fact that they were introduced into the Hudson Valley was, well, lucky for me that I was in the Hudson Valley to study them, because, you know, if you go north, farther up the Hudson Valley, you can see there's no point there showing, and that's because they're probably just not even there, because they're very localized, which is strange, and I'm not entirely sure why that is, but they're like distinct populations. So that's why I was also able to find the worms in one section at Bard, and then like a kilometer away, there just like were no worms. So that's another weird way that they behave. They're weird.
But I think really interesting. I mean, to hear about this. Does anyone else have any further questions for Wallis? We'll give it a moment just in case. Okay, I'm gonna not only unmute myself, but I'll also start the video again. Oh, what is the likelihood of jumping worms making their way to the Pacific Northwest? I mean, if they were in soil, they could do it, right? I mean,
yeah. No, I mean, I, have thought about that. Like, I mean, I thought about that a lot when I did my senior thesis and went home, because I did it over the summer. And then I went home to Washington for August. And so like, as I boarded the plane wearing like boots that I had been wearing around, even though I had like really brushed them off, I was like, what if I'm bringing them with me? So I'm not entirely sure. I don't know why they haven't really spread over there yet, considering that from my research, it seems like, and just how often I encountered incidences of them, despite their locality in the Northeast. I don't know why they haven't come over yet, but, and this is like kind of funny because it's ominous, but also like very like small, is that they move 6 meters a year. So, I mean, that's not like that. It's small. It is significant, especially for a worm, but each year, like a colony of them, a group of them, and the cocoons they lay, they can progress 6 meters in any direction per year. So, I mean, who knows? Maybe they'll inch their way to the Pacific Northwest, 6 meters a year.
Coming soon to a forest near you.
Yes.
So Jean says, looks like right now the Mississippi River is stopping them. So I would imagine, that there are a few barriers. But I mean, when we talk about how widespread trade is, people, I mean, it just seems like it's going to happen, right? I mean, it is in the cards, most likely.
Yep.
And obviously they'll show up in more places around us so that we might start finding them in our own backyards. I mean, you mentioned the fishing thing.
Yeah.
And when I think about the number of lakes and rivers in the Northeast, I mean, well, there you go, right?
Yep. No, for sure. Oh.
Okay. Are there any final questions before we let Wallis go? I want to thank everyone for joining us, and I want to thank you, Wallis, for sharing information that I had no clue about any of this stuff. For those of you who attended, tomorrow you'll be getting an email thanking you, but also asking for any feedback. So if you have any suggestions, things you liked, things you didn't like, topics you're curious about, please send them to me. My email will be there, mzaplatel@starhop.com. If you get out of this and then you suddenly have a bunch of questions about invasive jumping worms, I am sure that Wallis would be happy to do her best to answer them. So you can also find her contact information at starhop.com if you go to who we are. And so you can definitely send her an email and I'm sure she'll get back to you as well. Just to let you know, we've already planned our next Superstellar Friday. So as a heads up, we're going to have a guest presentation from the Mount Washington Observatory, and they're going to be talking about the science of nor'easters. So once I have that all set up, there'll be a registration link just like this. So keep an eye out for that one. But thank you again for joining us. Hope you guys have a wonderful evening and a great weekend as well. Enjoy the beautiful weather outside if you're near us, because today was pretty fantastic, and I'm really looking forward to tomorrow as well. Thank you to everyone, and thank you to Wallis. That was great, and I loved the video. I mean, it was kind of freaky, but I loved the video.
No, everyone is always like, how can you pick that up? I'm like, I don't know. It's one of the only bugs I can deal with, or well, not bugs, but creepy crawly I can deal with.
Yeah, but then you told the story about feeling the tail and fall off.
Yeah.
And that totally changed my opinion in general about these
things.
things.
Yeah. No, I mean, I don't think I ever want to pick up a worm again, but.
Sounds good. Wonderful. So Wallis, you are the host. So if you would please.
Oh, okay. Yes. All right. Goodbye, everyone.
Bye.